In this video, we will explore how to build a metazoan. There are thousands of species of single-celled eukaryotes. Lineages of photosynthetic eukaryotes are loosely grouped as algae. Several heterotrophic eukaryote lineages are grouped as holozoans, which are a group of organisms that includes animals and their closest single-celled relatives, but excludes fungi. Others are grouped as fungi. These groups of eukaryotes diverged at some point in the Proterozoic. I have mentioned the term metazoan multiple times, but what are they and how do we make one? Metazoans are not just multicellular animals. They are multicellular animals that have different kinds of cells that perform different functions. They most likely evolved once, and they share the same kind of early development. There are three basic body plans for metazoans: sponges and their relatives with no symmetry, cnidarians and their relatives with radial symmetry, and the bilaterians, which are 3D animals with distinct bilateral symmetry. Sponges are the simplest metazoans living today. They contain many flagellated cells called choanocytes, arranged so they generate efficient feeding currents. The cells are linked firmly together to form a body wall with no symmetry. You can completely disaggregate a sponge into individual cells, and multiple new sponges will grow because they are built on a cellular level. This illustration shows the three major sponge structures. They are basically modular filtering units. The choanocytes are red, and the outer skin is yellow. The blue arrow denotes the outgoing water current as they feed. Cnidarians include sea anemones, jellyfish, and corals. They are more complex than sponges and are built mostly of sheets of cells. They exploit the large surface area of these sheets in sophisticated ways. They contain a lot of seawater in a largely enclosed cavity that's lined by the inner surface of the sheet. This is the basic body plan of a cnidarian. They have a two-sided sheet of tissue that defines the inside and outside surfaces of their bag-shaped digestive cavity. They all have tentacles, a mouth, and a digestive cavity. They all have radial symmetry that radiates outward from the mouth as the central point. In most cnidarians, the outer surface of the sheet forms a protective skin, while the inner surface is mainly digestive. They can exist on small amounts of food and absorb oxygen from the water that surrounds them. They have nematocysts, which are stinging cells set into the outer skin surface. These cells are an effective defense and powerful weapons. The third and most complex group contains all other metazoans, including the vertebrates. These are the bilateria or bilaterians, which are metazoans with a distinct bilateral symmetry. The front end of bilaterians features food intake, a mouth through which food is passed into and along a specialized one-way digestive tract. They have a circulatory system and an internal fluid-filled cavity called a coelom, which may have evolved as a useful hydraulic device. Many advanced bilaterians have segments. Their bodies are divided by septa that separate the coelom into separate chambers connected by valves. This makes their bodies efficient for burrowing. Many bilaterians that live in shallow burrows have various kinds of tentacles, filaments, and gills that extend into the water as respiratory or feeding organs. These two worms, the feather duster worm on the left and the Christmas tree worm on the right, are marine bilaterians that build tubes as their own burrows. From their tubes, they extend tentacles to collect food and absorb oxygen from the water. Before we adventure further, we need to define the major marine habitats because much of the life we will be discussing for the next few 100 million years are all marine. There are two main oceanic life zones, the pelagic zone and the benthic zone. The pelagic zone is the open ocean waters. Life in the pelagic zone is divided by how they move around. Plankton are passive floaters, such as diatoms, foraminifera, and radiolaria, or weak swimmers, such as copepods, larval fish, and jellyfish. Nekton are strong swimmers, including most fish, squid, turtles, and marine mammals, for example. The benthic zone is the sea floor at all depths. Life in the benthic zone lives either on the ocean bottom or within sediment deposits and may attach as sessile benthos or burrow or move freely as vagrant benthos. If benthic fauna live on the ocean bottom, they are called epifauna. If they live within the sediments, they are called infauna. Examples of benthic fauna include sea stars, crabs, worms, clams, sea cucumbers, sea anemones, urchins, snails, corals, and barnacles. Looking at this illustration, you can see how easy it is to build a complex structure by careful placing of identical, simple units. Metazoans have structural genes to build each piece of the animal, and regulatory genes that make sure each piece is built in the right place at the right time. Homeobox genes, or HOX genes for short, are sets of regulatory genes that sit close to one another in the DNA of metazoans. HOX genes control the growth of nerve nets, segments, and limbs throughout the metazoans. They provide separate but complementary evidence to help read the evolutionary history of metazoans, and in early metazoans provided a genetic toolkit to guide the construction of viable, complex animals. When one animal group is radically different from another and is considered to be a group that evolved from some single ancestral species, it is a phylum, defined by its own particular body structure, ecology, and evolutionary history. Arguments about the number of phyla among living metazoans are ongoing between biologists and paleontologists. There are a bewildering variety of worm-like organisms, for example, that do not typically exist in the fossil record. Most biologists would suggest about 30 phyla, but only 7 phyla and 3 classes are or have ever been important in paleontology because they have hard parts that fossilize well. Advanced bilaterians form 3 major clusters of phyla. The ecdysozoa are animals that molt off their outer skins or outer skeletons as they grow. These are primarily the arthropods, which includes the extinct trilobites, crustaceans, and insects. The Lophotrochozoa are animals with small floating larvae and a way of life that involves filter feeding from the water with a tentacular or filamentous organ called a lophophore. They include the brachiopods and the bryozoans. The third cluster of advanced bilateria phyla are the Deuterostomia, which are originally filter feeders with floating larvae, but the larvae are so different that they cannot belong to the same clade. They include the Echinodermata, which are sea stars and their relatives, and the Chordata, including humans and minor related groups. The defining characteristic of the deuterostome is the fact that the blastopore becomes the anal pore, and that is the first thing to typically form in a deuterostome. Shortly before the Cambrian period, around 555 million years ago, the bilaterian groups diverged, but in ways that left no significant record of body fossils. Finally, at the base of the Cambrian, groups split into phyla, many with hard parts, which left a rich fossil record. This is the end of part 2. Next, we will investigate the Metazoan radiation and the Cambrian explosion.